Hello and welcome all in this video. So we are going to discuss about the mail report system of the prawn. So we have to discuss in this video about the mail report system of the prawn. So in case of prawn, there is the sexes are separate. That is the dioecious condition is found in the prawn. That is sexes are separate, and also sexual dimorphism is well marked in the prawn. That is male is bigger than the female and possess narrower abdomen, the male having narrower abdomen than the female, and the second ciliate legs of the male is longer, stronger, and more spiny. Also, in case of male, the pair genital opening lie on the cocci of the fifth pair of walking legs, while in case of female, paired genital openings lie on the cocci of the third pair of walking legs. With the help of these characteristics, we can differentiate male from the female. That is, the male is bigger and possesses narrower abdomen. Second ciliate legs is larger, longer, stronger, and more spiny. In case of male, the paired genital opening is found on the fifth pair of walking legs. And in case of female, the genital opening lie on the cocci of the cocci means the base of the third pair of walking legs. About the male reproductive system. So in case of male reproductive system of the prawn, there is having a pair of testes. There are having the two testes. The testes are soft, white, and elongated bodies which fuse at their anterior end. These testes are fused at their anterior end and to form a common lobe. But they having a gap. They leave a gap between them, and that gap is called as gap for cardiopyloric strand. This gap for the cardiopyloric strand, this gap, in that gap, there is a connecting by this gap, gap of cardiopyloric strand, the cardiopyloric strand connecting heart to pyloric stomach. By this gap, the heart is connected to the pyloric stomach. So in between the testes of the prawn, there is a gap for the connection between the heart to the pyloric stomach. Next part of the male report system in case of prawn, that is having a vas difference. That is the vasa differentia or vas difference. This vas difference is a long, coiled, and narrow tube, and arise from each testis. Arise from the each testis, and on emerging out, at the site of emerging out, the vas difference of each side at once to form a much coiled mass, and then runs vertically downwards between the abdominal flexure muscles on the inner side and thoracic wall on the outer side. That is, at the time of connection with the testes, this vas difference forming a coiled structure, and that is called as the mass of vas difference. Again, when this vas difference tube goes towards the posterior side, becomes straight and then open into a particular type of chamber that is called as seminal vesicles or vesicula seminis. So the each vas difference reaching ventrally near the base of the fifth walking legs swells to form a club-shaped vesicula seminalis or seminal vesicle. And this is the place where the spermatozoa is stored in the form of white compact bodies. So seminal vesicles then again open to the exterior, open to the outside by a particular pore, and that particular pore is called as male genital pore. And in case of male, this genital pore is found at the base or the cocci of the fifth pair of walking legs of each side. Each male genital aperture is covered by a small flap of integument. So these are the structures which are found in the male report system. In case of prawn, that is having a pair of testes, pair of vas difference, pair of seminal vesicles, and pair of male genital pore. When we see histological structure of the testes, we can find that histologically each testis consists of large number of coiled tubes, and that is their tubes are called as tubes are called as seminiferous tubules. So internally there is having many tubes in that particular testis, that is called as seminiferous tubules, and the seminiferous tubules embedded in the connective tissue. These tubules are then connected with the connective tissue, and where there in these tubules, in this seminiferous tubules, the process of spermatogenesis takes place. In this seminiferous tubule, the process of sperm formation takes place. The process of formation of sperm is called as spermatogenesis. So sperm formation or spermatozoa formation takes place in these seminiferous tubules. In the seminiferous tubules, the sperm formation processes continue or takes place. When we see the mature sperm having two main structures. The mature sperm consists of a rounded cytoplasmic body and containing a large dark nucleus in the head portion and a tail-like blunt process. So when we see this mature sperm, we can find they are having a head, rounded head, and having a large nucleus and a tail-like process in the sperm. So this is all about the male reproductive system of the prawn.